It's a real pleasure to be here. I'd like to give the last talk in this insightful morning on a global aspect of regulation to answer three important questions. Why is regulation so important for the development of the human infant? What is the stage-by-stage development of regulation from birth to adult life? And what are the specific environmental provisions required to sustain it? But before I start, I must say a word of thank you. I have been here in the think tank since the second think tank, which was merely 2 years ago. We were 35 people in Vicky's living room. It's amazing to see how exponentially this has grown in just 2 years, in size, in volume, and particularly in impact. It's amazing to see how one woman with a vision, with determination, with endless energy, with a tenacious belief in the first years of life, and with great generosity, a real heartfelt generosity, can make such an impact on so many children. And us scientists who study infants and their developing brain, and those who come in contact with families and treat or care for infants, and on those who make policies to shape the environments where children grow. So thank you very much, Vicky. You are a true inspiration. That said, let's go to regulation. Mammalian young are born with two important constraints. The brain is immature at birth, and young maintain close proximity to a nursing mother, mammals. Regulation is just defined as the process by which the immature physiological and behavioral systems of the young mature, organize, and adapt to the living ecology via physical contact with a caregiving adult and ongoing social exchanges that are well-timed and adapted. There are therefore three lines in the development of regulation. The first is the maturation of the organism, or self-regulation. The second one is the co-regulatory context, which is synchrony. And the third is the evolutionary timeline, how immature the brain is at birth. And we human, the apex of evolution with the most exquisite brain, we have the slowest maturity. Our brain is the most immature at birth and requires the longest period of dependence on the co-regulatory context. These three lines are interdependent and are supported to some extent by the ancient oxytocin molecule. The oxytocin molecule evolved presumably approximately 650 million years ago via gene duplication in jawed fish. It is implicated for the regulation of basic life function, such as energy balance, water conservation, thermoregulation. However, within each species, oxytocin has been repurposed in support of social life. So it became implicated in social functions, such as parent-infant attachment, pair bonding, social collaboration. And thus, within each species, within each organism, this charted the mechanism that stresses are regulated through social contact. So whether you are a very simple organism or a very complex one, social function, parent-infant attachment, and social bonding is the way that helps organism manage the stress involved in living in harsh ecologies. So if we want to go to a model of the development of regulation, in the newborn period, developments that occur in the third trimester of pregnancy are responsible for the development of brainstem-mediated periodicity, regulation of sleep-wake cyclicity, regulation of heart rate variability, regulation of state control. In the first year of life, between about 2 months and about 12 months, emotion regulation kicks in. Infants learn a repertoire to manage heightened states of positive or negative emotion. In the second year, following the first maturation of the prefrontal cortex, attention regulation. Infants acquire mechanism to redirect, focus, and maintain attention. Those lead in the preschool years with the development of the rudimentary sense of self to use that self as a coherent structure that regulate all the social and emotional experiences a child encounter. And this sense of self expands in culturally adapted way throughout life. We learn to regulate in more and more complex ways, both physiological, emotional, and attentional experiences. The parental function that regulate each stage of this model, maternal or parental physical proximity regulate the physiological, brainstem-mediated regulation, synchrony and attachment regulate help regulate the emotional component, parental scaffolding, symbolic play, and language help regulate children's attention, and parents' warm plus control parenting and narrative expansion help children expand their sense of self within cultural meaning system. As we began to follow children in 6 or 7 cohorts from birth to adolescence, we began to put together the puzzle of how self-regulation and how synchrony develop in a step-by-step way from birth to adolescence. Important to say is that what initiates this process, what jumpstarts it, is the tremendous alteration in the parental brain in mothers through pregnancy and childbirth, and in fathers through the first weeks of life through active caregiving, which through brain-to-brain synchrony with the infant set this two-brain experience by which the parent's mature physiology externally regulate the infant immature brain. So let's go to the specific. Newborn physiological regulation. Newborns learn to regulate their sleep-wake cycle, heart rate variability, and state regulation. In the first year of life, infants acquire a regulatory repertoire, maintain balance during stress, and learn to return to baseline following a stressful experience. In the second year of life, infants learn to maintain focus on task, goal-directed attention, and tolerate the frustration that are involved in learning. In the preschool year, infants acquire a rudimentary self-concept. They learn emerging morality, right and wrong, and begin to acquire self-restraint. Those give rise in adolescence to a host of positive outcomes: empathy, executive functions, the ability to manage stress and dialogue conflict, behavior adaptation, and lower accident proneness. Now, when we looked for how those different stages of regulation support, buttress all these competencies in adolescence, we found 2 mechanisms. One is step-by-step continuity. You do better physiological regulation, you're better prepared, you'll have more emotion regulation, you'll then enter the next stage with better a capacity for attention regulation, better self-regulation, and then better adolescent. But also another mechanism that works above and beyond this gradual continuity and really speaks to the goal of the think tank is this direct continuity, so that there is a direct prediction from what happened in that sensitive period to adolescent outcome, as if this early experience create this neurobiological milieu of a brain that is more regulated and is more fitted to the social context to develop later on better in adolescence. What is also important to say is that each form of regulation builds upon and integrate earlier forms. So when you're an adolescent, of course you have to regulate your physiological processes and emotional processes and attentional processes, but you have more refined, more complex, greater repertoire of strategies to do that. Biobehavioral synchrony, our second line of regulation, is defined as the coordination of biology and behavior between affiliated members during moments of social contact. So we found that when parent and infant, both mother and father, coordinate synchronous nonverbal communication signals with their infants in the gaze, affect, vocal, and touch modality, there's also coordination of heart rhythms, which appear during moments of synchrony and do not appear during moments of non-synchrony. There's a coordinated release of oxytocin, and there's brain-to-brain synchrony in key nodes of the social brain. We followed these sample. This is my longest sample that I've followed, my PhD research, from birth to now 22 years. And we see that those motives, those ritualistic synchronous interaction between each parent and their child are diet-specific. And like the 12-bar blues in jazz, they keep on playing throughout the interaction where more and more complex skills kick in. But when you see the interaction at 3 months, at 14 years, and at 22 years, you find the same motive operating in it again and again. This is what gives that interaction the secure base. Synchrony develops. At newborns, there is the expression of the parent species-specific repertoire. In the first year of life, there's a coordination of gaze, affect, vocalization, and touch. In the second year of life, parents tunes and expands the children's symbolic expression. And in the preschool years, the parent elaborates the child imagination and narrative. And again, this gave rise to exactly the same outcomes in adolescence, using exactly the same mechanism of step-by-step continuity. You're more synchronous at one stage, you'll develop more mature mechanism to express synchrony and intimacy in the next stage, but also a sensitive period perspective that greater synchrony in the sensitive period of birth to 9 months predict adolescent outcome directly, refocusing our work to the first year of life. And again, each form of synchrony expands and integrates the earlier form. Here is one more important to say about synchrony is that synchrony doesn't only go lengthwise from birth to adult life, it also goes widthwise across human attachment relationships. So those 4 forms of synchrony in the behavior, in the heart rate, in the endocrine fate hormonal release, and in the brain-to-brain synchrony are found. They are established in the parent-infant relationship during the early sensitive period, but they appear again and again in all 4 forms of human relationship. Parent and infant, close friendships, romantic attachment, and our relationship with humankind. So humans are the only species that we can feel empathy, activate the neural circuits of empathy, and release oxytocin both to the distress of a stranger and to the experience of shared collaboration with strangers. So this is a study when we followed infants 7 times across the first 10 years, and at each point we measured both synchrony and emotion regulation. And at 10 years, we measured various regulatory outcomes. And we found that every step of the way, regulation was stable, and synchrony was stable. You're more regulated at 3 months, you'll be more regulated at 6 months, you'll be more regulated 12 months, you'll be more regulated at 24 months, etc. But there is also cross influences. So children who were more regulated at birth had better synchrony with their parents at 3 months, leading to better regulation at 6 months, to better synchrony at 1 year, and vice versa. So that field of mutual influences between the child's inborn disposition and the parent's synchronous interaction is that mutual field that builds in a diet specific to that parent and infant, this field where the infant self-regulatory capacities mature within the co-regulatory context. What jumpstarts that? What is the fundamental biological change that starts this whole process of self-regulation and synchrony? Is the alteration of the parental brain. It's the parental caregiving brain. Now think that adult neuroscience has shown us that the immediate pre- and postpartum period is the period that is the most plastic in the adult brain. So if the human brain is the highest achievement of evolution, and the postpartum period is the most plastic period of that brain, we're talking about the parental brain as the highest achievement of the entire evolution processes. What we found when we look at fMRI, and we summarized all 46 studies up to date that did fMRI studies of parents exposed to their infant stimuli, that the parental caregiving network, that parental brain, consists of several networks that cohere into the parental caregiving network. The first network is a subcortical network that is deep in the brain, includes the hypothalamus that produces oxytocin, that connects to the amygdala to care and be vigilant for your infants, and to the VTA, the dopamine reward circuitry. And this is a network that we share with all mammalian mothers, rodent mothers, primate mothers, and human mothers. But in human, this evolutionarily conserved mammalian caregiving network is connected via multiple ascending and descending projections with several cortical systems that enable human parents to really parent in a human way. So we are including the empathy network, insula cingulate circuits that enable parents to feel infant pain and emotions within their own brain, and through von Economy neurons, ground experience in the present moment. The mirror neuron system that enable us to represent other actions in our own brain. The mentalizing network that enable us to understand infants' intention through their action. This is the theory of mind network. And the latest evolving emotion regulation network, more frontal structure, that enable parents, human parents, to plan future parenting, multitask, and direct parenting according to culture-specific goals. In a first study that we did on the parental brain, we wanted to see how synchrony is implemented in the parental brain. So we had 45 mothers. We went to their home. We wanted to see them in the natural ecology. And we microcoded their behavior. And we divided them to two groups. Mothers who were synchronous, that coordinate their interaction with the signals of the infant, and mothers who are intrusive, those who overstimulate when the infant has signals a need for rest. And then we exposed all mothers to their infant stimuli. We show these interactions to the mothers as when they were in the fMRI, in comparison to strange mothers and their infant, in order to target how the brain perceives attachment-specific experiences. What we saw when we differentiated which structure differentiated between the synchronous and the intrusive, we saw two structures, both from the subcortical mammalian general network. One was the amygdala, the vigilance, which was much more active in the intrusive moms, and the synchronous moms had much greater activation of the nucleus accumbens, which is a critical node of the dopamine reward circuitry. We then did functional connectivity throughout the entire movie. So we wanted to see what works alongside the amygdala, what works alongside the nucleus accumbens. And we found that for the synchronous mothers, along the nucleus accumbens, the reward underlying coloring, there was activation of all circuits of the parental brain. In contrast, for the intrusive moms, Alongside the amygdala, there were only some sensory motor activation, nothing of the empathy, resonance, embodied stimulation that help mother represents the infant needs in her own body. Synchrony is then represented in the mother's brain through an underlying reward coloring and integration of the entire cortical network that enable mothers to feel, read the intention, and plan for good parenting, all those connected with a color of reward in the mother's brain. Next, we wanted to see is, does activation in the parental caregiving network bears any consequences for the development of infant social and emotion regulation? Is this what's going on in the brain? I'm just showing you nice colors, or it has any implication for children's development? So we followed children. We had 91 parents. Some of them were mothers, some of them were fathers, and some of them were gay fathers who raised infants in partnership with no maternal involvement. So we had some mothers who were primary caregivers, some fathers who were secondary caregivers, and some fathers who were primary caregivers. We came to the kids' home when they were 4 years of age. And we measured socialization through a pickup task. And we had regulation, two paradigms that measure the regulation of negative and positive emotion. So we had the fearful mask. The experimenter wears various masks. And then we had the bubbles. It's like a joyous experience to elicit positive emotion. And we microcoded both the expression of negative and positive emotion and the type of strategies children, preschoolers use to regulate those emotions. Now, we took a network cohesion approach to the brain structure. What it means is that we looked how structures which within each network cohere to function together. So we used an algorithm that measured network coherence in the mammalian subcortical network, in the embodied simulation network, which combines the empathy and mirror network, and in the mentalizing theory of mind network. So here we see how connected those structures were in the parental brain during the sensitive period of infancy. So this is from infancy to the time children are 4 years old. What we found is that children who had parents with stronger connectivity of the subcortical mammalian general network expressed more positive emotion when they were 4 years old and used simple regulatory strategies to regulate positive moments, such as laughing or physical proximity seeking from the adult. Children whose parents had greater connectivity of the embodied simulation network were able to use more complex regulatory strategies to regulate negative emotions, such as symbolic play or attention diversion. And children whose parents had greater connectivity of the mentalizing network were better able to self-regulated compliance when they were asked to pick up the task. At 6 years, children whose parents had greater connectivity of the embodied simulation network had lower cortisol production, and this is similar to other mammals, that functionality of the parent brain in the postpartum predict the child's lifetime ability to manage stress. And very interesting that the link between the parent embodied simulation connectivity and self-regulation strategies at 6 years pass through parent-infant synchrony. So the embodied simulation network, which enable humans to feel the emotion and actions of other in their own brain. We can represent what you are experiencing in my own brain, the system that implicates intersubjectivity, combines with social synchrony to tune the infant's brain into the social world. I'd like to very quickly describe to you two MEG studies, MEG studies, one that shows brain-to-brain synchrony between mother and child, and one that shows outcome of postpartum maternal depression in children who are adolescent. The first study, we took mothers and young children, and we videotaped them in the home ecology, and then mother was in the MEG, child was in the MEG, and they watched that movie. Okay, we wanted to see whether their brains synchronized. So the first thing, mother and child in the MEG observed their own interaction. They observed unfamiliar mother-infant interaction. We coded the interaction for episodes of synchrony, gaze and affect synchrony, and then we measured brain-to-brain synchrony. We had 3 very interesting findings. First of all, we found that mother and her own child, when they observe their own interaction, not when they observe an unfamiliar interaction, they synchronize their brain in the STS. The STS is a key area of the social brain that has overlapping mentalizing and mirror properties, both theory of mind and mirror functions. The second thing we found is that this brain-to-brain coordination between mother and child was expressed in gamma oscillation, fast-paced rhythm that implicate bottom-up behavior-based mechanism. And the third thing that was really interesting is that mother and child coordinated their brain response only during periods of synchrony. When they watched synchrony, their brain synchronized. When there was no synchrony, their brain didn't synchronize. This tells us that condition of high and toxic stress that really reduce the amount of mother-infant synchrony, such as anxiety, such as depression, such as trauma, really do not enable brain-to-brain synchrony between mother and child. And the last data, this is a longitudinal study from birth to adolescent. Mothers were depressed throughout the child's first years of life. At 12 years, children were exposed in the MEG to painful stimuli, pain versus not pain, individuals in painful versus not painful. And we wanted to see how their brain responds to the pain of others. What we found is a difference between children growing to depressed and non-depressed mothers. And the difference is, again, in that structure, the STS, which combines mirror and mentalizing property. Children of depressed mother aborted processing of theory of mind, did not process, shut down the processing in the social brain when they saw the pain of other. And this is not because they were not able to identify that the other is in pain, but because the experience was so overwhelming that they just couldn't have their brain, so they terminated brain activation, the brain processing of this experience. Just to summarize, synchrony is the process by which the physiological and behavioral system of one human coordinate with those of another. And this process provides the basis for physiological, emotional, and self-regulation. Such model of synchrony may provide a window to addressing the subjectivity gap, the key question in current neuroscience, how a model of the human brain can address experiences of the human mind. This is my students. Thank the students who carried out this study. Thanks to our funding agencies, and thank you for listening.